The story begins in 1869 with Friedrich Mescher, who isolated the chemical nuclein from pus collected. We know today that genes are sections along the DNA molecule of chromosomes and that genes specify the instructions for the synthesis of proteins. But this information pathway was unknown up to the middle of the 20th century. It is instructive to look at how the information pathway that we call the central dogma today, that is, DNA to RNA to protein, was discovered. The story begins in 1869 with Friedrich Mescher, who isolated the chemical nuclein from pus collected from hospital bandages. Mescher found that nuclein was a component of the nuclei of white blood cells, the infection-fighting cells of the immune system. Although Mescher knew that nuclein was rich in phosphorus and behaved as an acid, he did not know that it was DNA. Around 1920, nuclein was shown to be DNA, the hereditary molecule that carries the instructions for protein synthesis. Scientists knew by the middle of the 20th century that DNA was found in the nucleus, but that proteins were synthesized in the cell's cytoplasm. How did the instructions in DNA direct the construction of proteins in the cytoplasm? It was hypothesized that a messenger molecule must be present which traveled from the nucleus to the cytoplasm. In a series of experiments, this molecule was found to be RNA, and was therefore called messenger RNA, or mRNA for short. This lesson will explain how genes are expressed by the mechanism of copying DNA into mRNA, a process called transcription. The information contained in the mRNA is then used to make proteins in a process called translation. 3 types of RNA are involved in transcription and translation. These are 1. Ribosomal RNA, or rRNA, is a component, along with proteins, of a complex molecular machine known as a ribosome. A ribosome is composed of 2 subunits, referred to as the small and large subunits. 2. Messenger RNA, or mRNA, is a single-stranded copy of the template strand of DNA. mRNA contains codons that specify the type and order of amino acids in the polypeptide. 3. Transfer RNA, or tRNA, is a cloverleaf-shaped molecule that carries an amino acid on one end and an anticodon on the other end. It transfers amino acids to the mRNA and ribosome complex. Complete the figure by dragging the descriptions and molecular shapes that match the 3 types of RNA. You have not finished this activity. Transcription involves the assembly of an mRNA molecule from a template strand of DNA. First, the enzyme RNA polymerase binds to a specific sequence of nucleotides called the promoter. Then, RNA polymerase unwinds the DNA double helix. This is the initiation stage. In the second stage, called elongation, ribonucleotides enter the catalytic site of the enzyme, and an RNA strand is built using one DNA strand as the template. The new RNA strand is complementary to the DNA strand because of the use of the base pairing rule. DNA and RNA use the same nitrogenous bases, except that RNA uses uracil instead of thymine, which is only found in DNA. Termination of transcription occurs when RNA polymerase encounters a stop signal within the DNA sequence. RNA polymerase then separates from the DNA strand. From the pool of ribonucleotides, assemble the next portion of the RNA strand by dragging the correct ribonucleotide to the appropriate position within the RNA polymerase. You have not finished this activity. Following transcription, the pre-messenger RNA transcript is processed in the nucleus to remove non-coding sequences called introns. The coding sequences, or exons, are those that will code for a protein. The exons are spliced together following removal of the intervening sequences, or introns. The processed mRNA enters the cytoplasm through the nuclear pores, where it binds to a small ribosomal subunit to initiate the translation process. A charged tRNA molecule carries the first amino acid, methionine, to the small subunit mRNA complex. The nucleotide code is read as a 3-letter codon, with the codon AUG signifying the start of translation. A large ribosomal subunit with 2 binding sites for tRNAs called the P and the A sites binds to the small subunit, which then forms a functional ribosome. The first charged tRNA binds to the P site, while the next incoming charged tRNA will bind to the A site. At one end of the tRNA molecule is attached an amino acid, while at the opposite end is the anticodon, a 3-nucleotide sequence that will bind to the codon if they are complementary. From the pool of charged tRNAs, select the one whose anticodon is complementary to the start codon, AUG, and drag it to the start codon on the mRNA. This begins the initiation stage of translation. You have not finished this activity. Elongation is the second stage of translation and begins by the ribosome moving down the mRNA 1 codon at a time. Another charged tRNA comes into position and binds with the mRNA codon through complementary base pairing of its anticodon. Drag the appropriate tRNA to the next codon on the mRNA. You have not finished this activity. The final stage of translation is termination. Elongation of the polypeptide chain is terminated when a stop codon is encountered. When the stop codon is reached, here it is UAA, translation ends by the binding of a release factor to the stop codon. The finished polypeptide chain is then released from the ribosomal complex. The ribosome separates into 2 subunits and another round of translation can be initiated.